Good morning everyone. Today I am going to discuss about the sequence analysis of protein. So what is meant by the term sequence analysis of protein? Protein sequencing, it is a technique to determine the amino acid sequence of a protein. It is a method which helps us to understand the structure and the function of proteins in a living organism. So what is the importance of amino acid sequencing? Knowledge of a protein's amino acid sequence is necessary for detecting its three-dimensional structure and is also essential for understanding its molecular mechanism of action. Okay, so protein sequencing, amino acid sequencing will help us to get an idea about the structure, overall three-dimensional structure and function of the protein. Okay, so there are many diseases which is caused by mutation, for example sickle cell disease. So this result due to amino acid change in the protein. So amino acid sequence analysis can help us in the development of diagnostic test and effective therapies and formulating effective therapies. So the first amino acid sequencing, it was done by Frederick Sanger, and the first protein to be sequenced is the hormone insulin, and it was done in 1953. So first I will give you an idea about the steps in sequence analysis. So first one is the preparatory stage. Okay, what happens here? We know that there can be several subunits in a protein. So this individual polypeptide, we have to determine the individual polypeptide chains. We have to determine the number of subunits in a protein and the disulfide bridges. That is the first step. And we want to separate and purify each polypeptide chain. This number of polypeptide chain is determined by treating this protein with dinyl chloride, that we will discuss later. Okay, so what happens after this preparatory stage? If the polypeptide chain is too large, we want to cleave into small fragments by using enzymatic or chemical method. And again we want to separate and purify all these small fragments. Okay, so what happens after that? Next step is the sequencing. Okay, sequencing, we got small polypeptide fragments, and there is an N terminal and there is a C terminal for each polypeptide chain, and the C terminal is identified by carboxypeptidase, and the N terminal is identified by there are three methods. First one is using dinyl chloride, second one is using Edman's degradation method, third one is by using Sanger's reagent. Okay, once we got the N terminal and C terminal, then we can identify then the entire amino acid sequence by using Edman's degradation method. Okay, so the entire amino acid sequencing is done by Edman's reagent, Edman's degradation method. So we got several overlaps of different sets of fragments, and complete sequencing is done by comparing the overlaps of different sets of polypeptide fragments. So these all are the steps in sequence analysis of protein. So we'll discuss in detail. First step is the obtaining pure sample of protein, that is by technique known as chromatography. Various types of chromatographic techniques are used for separation, for example ion exchange chromatography, adsorption chromatography, high performance liquid chromatography, etc. Once we got a pure sample, the purity is assessed by electrophoretic methods such as agarose gel electrophoresis, polyacrylamide gel electrophoresis, etc. So once we got a pure sample of protein, we want to determine the number of polypeptide chains in a protein, and if protein contains multiple subunits, the number of polypeptide chains is assessed by treating that protein with dinyl chloride. So this dinyl chloride reacts with the polypeptide chain to form dinyl polypeptide, and upon acid hydrolysis, dinyl amino acids are released. So from the number and nature of the dinyl amino acid, we will be able to determine the number of subunits in a protein. So the number of polypeptide chain in a protein is detected by treating that protein with dinyl chloride. So if the polypeptide chain is too long, then it has to be broken down into small fragments by enzymatic or chemical cleavage. The common enzymes used are trypsin, chymotrypsin, etc., and the chemical is cyanogen bromide. Next, it is the end group analysis. There is an N-terminal end, there is a C-terminal end. Terminal end has been identified by treating with Danisel chloride or Sanger's reagent or by Edman's degradation method. So, if we use Sanger's reagent is 1-fluoro-2,4-dinitrobenzene that react with the amino nitrogen of N-terminal amino acid form a derivative of N-terminal amino acid. And upon acid hydrolysis, it cleaves all of the peptide. This acid hydrolysis cleave all of the peptide bonds, leaving a mixture of amino acid, and only one of which it bears the 2,4-dinitrophenyl group. Okay, so this upon acid hydrolysis, it cleaves all of the peptide bonds, leaving a mixture of amino acid, only one of which bears the 2,4-dinitrophenol group. That must be the N-terminal amino acid. So that N-terminal amino acid which bears the 2,4-dinitrophenol group, that is identified by chromatography. But what is the disadvantage of this technique? The remaining peptide is totally degraded by acid hydrolysis. That is the disadvantage of this technique. So, we cannot repeat the cycle with the same sample. So, for second amino acid sequencing, we require a new stock of protein sample. So, what is the disadvantage? The protein, the entire polypeptide chain is hydrolyzed by, is degraded by acid hydrolysis. So, we only able to know the N-terminal amino acid. We will only be able to identify the N-terminal amino acid. The rest of the polypeptide chain is fully degraded by acid hydrolysis. So, for second amino acid sequencing, we need a new stock of protein sample. Same is with the Danisel chloride. There also the peptide chain is fully degraded by acid hydrolysis. So, we will be only able to detect the N-terminal amino acid. Next is the Edman's degradation technique by using phenyl isothiocyanate. Okay, so the Edman degradation procedure, it labels and removes only the amino terminal residue from a peptide. The N-terminal residue is labeled and removed from the peptide, leaving all other peptide bond intact. But compared to compared to Danisel chloride method or Sanger's reagent, here the peptide bond remains intact. So we can repeat the cycle with the same sample. Okay, with the Edman's with the same reagent. Okay, but what happens in what happens if you use Sanger's reagent or Danisel chloride method? There the peptide chain is fully degraded by acid hydrolysis. But here in Edman's degradation, all the peptide bonds remain intact. So the peptide is reacted with phenyl isothiocyanate under mild alkaline condition that converts the N-terminal amino acid to a phenyl thiocarbamoyl adduct. So here you can see the phenyl isothiocyanate that react with the amino nitrogen of N-terminal amino acid and the PTH derivative, that is phenyl thiohydrantoin derivative, that is isolated and identified by chromatography procedure. This PTH amino acid, the N-terminal amino acid, is isolated and identified by chromatography procedure, and the remainder of the peptide, that is subjected to second Edman degradation using the same reagent. We can use the same peptide. Okay. That is the advantage of Edman's degradation over other two. Then C-terminal amino acid can be identified by carboxypeptidase A and carboxypeptidase B. So the final sequencing is by Edman's degradation using phenyl isothiocyanate. This reagent forms a covalent bond to the N-terminal amino acid and it then reacts with the second amino acid and is useful for 10 to 30 amino acids. So the complete sequencing is done by comparing the overlaps of different set of fragments. Each peptide is analyzed and whole sequence of the polypeptide is determined as if fitting in the parts of a jigsaw puzzle. So here this is the Edman's degradation technique. The N-terminal end is labeled with the phenyl isothiocyanate and the N-terminal amino acid is released. Remaining peptide is again labeled with Edman's degradation technique. Again the same reagent phenyl isothiocyanate used, that is released, and the step is continued until the entire sequence is identified. So the steps in nutshell. Okay, so first the protein sample is purified, and then using dinyl chloride we identified the number of polypeptide chains, and we hydrolyzed it to small peptides using chemical or enzymatic cleavage, and then we analyzed the N groups, N terminal and C terminal and using carboxypeptidase A and B, N terminal using 3 method, dinyl chloride, then Sanger's reagent, Edman's degradation, and the entire protein is sequenced using Edman's degradation method, and the complete sequencing is done by comparing overlaps of different sets of fragments. Next is the Ingram's fingerprinting method. It is a simplified method and it detects the abnormalities in primary structure. You just study the name of the techniques. Ingram's fingerprinting technique. Next, other techniques such as tandem mass spectrometry can also be used to sequence the protein. So this is the steps in mass spectrometry. The proteins are extracted from cells and digested into small peptides. The peptide mixture is separated using chromatography, and the peptides are ionized and analyzed using tandem mass spectrometry. This is another method for protein sequencing. Next, the higher levels of protein structure. Here we have studied, till now we have studied about the primary structure of protein. The higher levels of protein structure can be determined by X-ray crystallography, UV light spectroscopy, circular dichroism, then nuclear magnetic resonance spectroscopy, etc. So, we have discussed about the steps in determining the primary structure of protein, and we discussed about the various techniques in detecting the primary sequence of protein and the techniques for determination of higher levels of protein structure. Okay, thank you.